In this lecture, we're going to discuss the process of the gene expression, and we're going to start with the central dogma of molecular biology, which postulates that the genetic information flows from DNA to mRNA to protein. The genes within the DNA specify the mRNA sequence, and the sequence of nucleotides in mRNA specifies the sequence of amino acids within the proteins. DNA is critical for cellular function, and the genetic information stored in it is essential for cellular function. When DNA is transcribed into RNA, it prevents any possible damage to DNA molecules. Essentially, these mRNA copies of the genes keep DNA intact. During transcription, one nucleotide is added to the growing chain of RNA per every nucleotide that is read in the DNA strand. During translation, every 3 nucleotides in RNA molecule match one amino acid in the sequence of a future polypeptide. For example, nucleotides 1 to 3 correspond to an amino acid 1, 4 to 6 designate an amino acid 2, and so on and so forth. In this illustration at the bottom, you can see that, for instance, this central codon, UUU, corresponds to a single amino acid, phenylalanine. The codon CJA corresponds to a single amino acid, arginine. Now, what essentially drives the gene expression in different cells? In addition to a variety of intracellular signals, you have to understand that a phenotype depends not on all the genes that are present in the genome, but on the genes that are expressed at any given moment. And this expression will change either due to the intracellular signals or due to environmental conditions that will alter the gene expression. We mentioned already that each amino acid is encoded by 3 nucleotides, something that is called a codon. There are 4 different nucleotides in mRNA: U, C, A, and G. Since every codon contains 3 nucleotides, there are 64 possible 3-letter combinations. It means that there are more codons than amino acids they have to encode. There are 20 amino acids that comprise the structure of the protein. Therefore, it makes the code degenerate or redundant. Degeneracy means that a given amino acid can be, not necessarily is, but can be encoded by more than one codon. And in this case, each codon always encodes only one amino acid. If you take a look at the table with the genetic code on the left, you can see that, for example, an amino acid proline can be encoded by 4 different codons. That illustrates the degeneracy of the genetic code. But at the same time, you can see that the codon CCU will always encode an amino acid proline and not anything else. The fact that each amino acid is encoded by 3 letters explains the influence of frameshift mutations on the protein sequence. If you will insert 1 or 2 nucleotides, that will affect the reading frame. For example, in this illustration at the bottom, you can see that you can go from right to left or from left to right, Not necessarily insertion, deletion would do the same, but if you add or remove one or two amino acids, you completely change the sequence of one or two nucleotides. You completely change the sequence of amino acids. Before the frame shift, this fragment of mRNA encoded amino acid serine, valine, proline, and tyrosine. But when you remove U and A, then the entire reading frame, which can be seen here, these are codon triplets, changes. Only the first triplet that encodes serine remains. Everything else becomes dramatically different due to the frame shift. However, if 3 nucleotides are inserted into the sequence of DNA or RNA, it simply adds an extra amino acid, but the rest of the protein, in terms of its amino acid composition, remains the same. Out of 64 codons, 3 are nonsense or stop codons. You can see them here. These codons signal for the termination of the protein synthesis, and as the result, the polypeptide is released from a ribosome. Protein synthesis starts at the start codon, which is AUG. It also encodes an amino acid methionine, and whether it serves as the start codon or as the codon for methionine is determined by the adjacent nucleotides, sort of the environment. So essentially, during translation, mRNA is translated by ribosome, which reads every 3 nucleotides until it reaches the stop codon when the translation ends. In this illustration on the left, you can see that all codons form 16 blocks. These blocks are specified by the first and second nucleotide. Each block can be further divided into a pyrimidine and a purine half. So pyrimidine half ends with U or C, while the purine half ends with A or G. You can see that some amino acids occupy the entire block of codons, while others get either pyrimidine or purine halves. We mentioned before that the genetic code is degenerate. It's quite important because it allows to reduce the possible negative impact of the random mutations. Amino acids that have similar side chains are usually encoded by similar codons. For example, aspartate and glutamate or glutamic acid are encoded by the codons which are very similar and differ only in its third letter. Therefore, if there is a mutation in the third letter, asparagine would be replaced to glutamate or vice versa, which is not particularly detrimental for the protein. In different organisms, genetic code is practically universal, which provides an unequivocal amount of evidence to the common origin of life on Earth. So now we're going to talk about each specific step, starting with transcription in prokaryotic cells. As we mentioned before, during transcription, the molecule of DNA serves as the template for mRNA synthesis. But in order to do so, double helix of a DNA molecule must unwind, forming what's called a transcription bubble. out of two strands that can be found within the transcription bubble, only the template strand is transcribed. In this particular illustration, the template strand is at the bottom. It means that molecule of mRNA is complementary to the template strand. But another strand that is complementary to the template strand is the top one, the coding or non-template strand. So again, the sequence of mRNA molecule is the same as the sequence of coding or non-template strand. The only difference is that while in DNA there's a thymine, T nucleotide, in RNA instead of T there is uracil, which just like thymine forms a pair with A with adenine. Now first, nucleotide at the 5' end of the synthesized mRNA molecule corresponds to the number 1 nucleotide on the DNA, so-called initiation site. When a nucleotide in mRNA or DNA are located before the initiation site, closer to 5' end, they are called to be located upstream. Nucleotides that are after the initiation site are downstream. It can be illustrated the following way. If this is mRNA molecule, this is 5' end, and this is 3' end, and this is +1, upstream, downstream. So what happens after the double-stranded DNA helix unwinds? Well, the enzyme that synthesizes RNA, so-called RNA polymerase, binds to the DNA. RNA polymerase consists of 5 polypeptide subunits, 2 identical alpha subunits, beta and beta prime subunits, and a special sigma factor. All 5 of them comprise a holoenzyme or the functional form. First 4 subunits, 2 alphas, beta and beta prime, form a core enzyme. The core enzyme assembles before transcription starts and disassembles after it ends. Sigma factor only participates in the transcription initiation. It facilitates the start of transcription from the appropriate initiation site. So how do we know that the initiation site is appropriate? Well, before a gene or a group of genes, there is a special non-coding sequence called a promoter. It's a sequence in the DNA molecule to which all transcriptional machinery binds. Once it is bound to promoter, it can initiate transcription. It's important to understand that promoters are critically important to determine whether transcription is constitutive or regulated, and we need to explain these terms. The term constitutive means that transcription happens all the time at pretty much the same level. The term regulated implies that there are certain factors, maybe environmental, maybe intracellular, that will turn the transcription on or off or somehow modulate its efficiency. two key regions within prokaryotic promoters are -10 and -35 upstream of the transcription start site. These regions, -10 and -35, are called consensus sequences. It means that they are very similar in all promoters, in different genes, and different species. The -10 region, also known as a Tata box or Pribnow box, contains the sequence TA-TA-AT, and you remember that TA reach sequences are usually easier to separate. -35 region contains the sequence TTG-ACA. These consensus sequences are recognized and bound by sigma factor. Once sigma factor binds to the promoter, the core enzyme can bind to it too. The AT reach region in the Tata box unwinds, and RNA polymerase produces first transcripts, so-called abortive transcripts, with a length of about 10 nucleotides, before the transcription essentially kicks off. The process of transcription itself, the process of RNA synthesis, is called an elongation. For elongation to start, RNA polymerase releases the sigma factor, and core enzyme then moves along the DNA template, transcribing the template strand into mRNA molecule. This mRNA molecule is synthesized in a 5' to 3' direction. Ahead of that transcription bubble, DNA is unwound, and it is rewound back behind the core enzyme. I can't stress it enough that out of 2 DNA strands, only template strand is transcribed, and a single-stranded transcript, single-stranded RNA, is produced. However, it's been demonstrated that in a cell, cellular RNA polymerases cannot transcribe a single-stranded DNA, and 2 strands of DNA are required all the time. Now, let's go back to our transcription process. How does it end? There are 2 types of signals to terminate transcription: protein-based, or Rho-dependent, and RNA-based, or Rho-independent. In Rho-dependent termination, a Rho protein, or Rho factor, moves behind RNA polymerase. It would be here. When RNA polymerase runs into a sequence of several G nucleotides at the end of the gene, it stalls. Rho protein that moves straight behind it hits the RNA polymerase, kicks it off DNA, and releases mRNA molecule. In Rho-independent termination, the process of ending the transcription is controlled by specific sequences in the DNA. For example, there is a region at the end of the gene, rich in G and C nucleotides. So when this region is transcribed, mRNA folds on itself, forming the intramolecular pairs between G and C. This structure is called a hairpin, and it would look something like that. And there's something else. So this is a hairpin. Then RNA polymerase, after the hairpin inside of it is formed, RNA polymerase starts stalling at the next region, which is AT-rich, and eventually thermodynamically core enzyme is released from the DNA, and mRNA transcript is released as well. So let's summarize the transcription in prokaryotes. It's interesting that the transcript, shown here in blue, can be used to synthesize the protein while the transcript is being synthesized itself. If you look at the illustrations on the right and on the left, the blue circular line represents the DNA located in the nucleoid region of the cell. When mRNA is synthesized, when transcription is actively happening, mRNA becomes available for ribosomes because there are no membranous organelles, there is no membrane that surrounds DNA, and minimal modification of RNA post-transcriptionally is required. In other words, this transcript, this mRNA transcript that I highlighted here, it is not yet completely synthesized, but it's already bound by the ribosomes, which start producing protein almost immediately. Now, you can imagine that this concurrent existence of transcription and translation in prokaryotes makes protein synthesis highly efficient and rapid. But not only this, you can see that each mRNA molecule is bound not by one, but by multiple ribosomes, so-called polyribosomes. Another important feature of prokaryotic mRNA is that it is often polygenic or polycistronic, which means a single transcript may contain multiple genes. Now, this simultaneous occurrence of transcription and translation in prokaryotes is unimaginable in eukaryotic cells because, first and foremost, the presence of a nucleus, the presence of a nuclear membrane, effectively prevents simultaneous transcription and translation because mRNA has to be exported from the nucleus into the endoplasmic reticulum or cytoplasm. Conceptually, eukaryotic transcription is similar to prokaryotic. However, there are some differences. So first of all, mRNA must be transported to the cytoplasm because eukaryotes have membrane-bound nucleus. They also must protect mRNA from degradation before it can be translated. Therefore, there is a requirement for some post-transcriptional modifications. Also, eukaryotes have 3 different RNA polymerases, and the mRNA that is synthesized during eukaryotic transcription is usually monogenic or monocistronic. In other words, every transcript contains a single gene. In addition to that, eukaryotic polymerase requires a certain number of transcription factors for the transcription to be fast and efficient. So 3 RNA polymerases are different due to their function. RNA polymerase 1 sits in the nucleolus where it synthesizes all of the ribosomal RNAs except 5S ribosomal RNA. The letter S, in this case, is the unit that describes how fast a particle precipitates during the process of centrifugation. This unit is called Swedberg. RNA polymerase 2 is fundamentally similar to RNA polymerase in eukaryotes, and it synthesizes all pre-mRNAs, all transcripts in the nucleus. RNA polymerase 3 synthesizes 5S ribosomal RNA and all transport RNAs. as well as small nuclear RNAs. Now, all this information is summarized in this table. I expect you to know the location of RNA polymerases 1, 2, and 3, and the product of their transcription activity. Similarly to prokaryotes, eukaryotic promoters contain Tata box, like in -10 sequence in prokaryotes. It's located on the coding strand. However, it is farther upstream from the initiation site, at location around 25 minus 35 bases. This box is also AT-rich, which facilitates the unwinding of the DNA template. Now, the promoter from RNA polymerase 2, they are usually pretty similar, but all promoters for RNA polymerase 2 are different from the promoters for polymerases 1 and 3. RNA polymerase 1, for instance, have 2 GC-rich promoter sequences in the -45 to +20 regions. RNA polymerase 3, on the other hand, contains promoters that are upstream of the initiation site, or sometimes promoters are located in the genes themselves. Let's dive further in the eukaryotic transcription. You may remember that prokaryotic transcription requires sigma factor. In eukaryotes, a similar role is played by transcription factors. Their role is to recruit RNA polymerase to a promoter. There is a set of transcription factors called basal, which bind to the promoter and recruit RNA polymerase 2. The fundamental basal transcription factor is TF2. There are several kinds of TF2 factors that have letter designations. The core complex of transcription factors consists of TF2D and Tata binding protein, or TBP. These transcription factors can later by others, and the whole complex of basal transcription factors is now functional to recruit RNA polymerase 2 and start the synthesis. Transcription factors help to increase the efficiency of transcription, but technically speaking, the transcription factors, many of them are not essential for transcription. For example, there is a bunch of transcription factors that bind to upstream regulatory sequences called enhancers and silencers. These sequences, along with the relevant transcription factors, regulate how much RNA is synthesized during transcription, but they are non-essential for the transcription itself. They're critical for regulating its efficiency, though. Transcription elongation in prokaryotes is very similar to the one in eukaryotes. However, you have to remember that in eukaryotes, genomic DNA is bound to proteins called histones. So these proteins have to be removed before transcription can proceed. Histone removal is the function of FACT protein complex, where FACT stands for facilitates chromatin transcription. FACT protein complex removes histones before the transcription bubble and returns them back after the transcription bubble moves farther. The termination of eukaryotic transcription relies on the temporary tail beyond the end of the gene, which facilitates the separation of RNA polymerase 2 from the DNA. and the release of the transcript from the enzyme. RNA polymerases 1 and 3 require special termination signals within the genes. As we mentioned before, in eukaryotes, mRNA requires an extensive processing to prevent its degradation and also to facilitate its binding to ribosomes. In addition to that, in the sequence of a gene in eukaryotes, there are two types of fragments, coding exons and non-coding introns. So these fragments that are interspersed through the gene have to be rearranged. Introns have to be removed before the translation, and exons have to be stitched back. It is conceivable also that the half-life of eukaryotic mRNA has to be longer than the one for prokaryotic mRNA, because it takes way more time for eukaryotic mRNA to be processed and exported from the nucleus into the cytoplasm or endoplasmic reticulum. Therefore, it must be stabilized. Stabilization is achieved by the use of RNA stabilizing proteins that protect transcript from degradation, and the processing that facilitates the binding of those proteins are 5' capping and polyadenylation. The intron removal is called splicing. In other words, mRNA processing in eukaryotes includes three main steps: capping, polyadenylation, and splicing. So what is capping? Capping is the addition of 7-methylguanosine to the 5' end of the pre-mRNA transcript. The role of 5' cap is to protect mRNA from degradation and to be recognized by translation factors to start protein synthesis. Polyadenylation is the addition of poly-A tail by the enzyme called poly-A polymerase. The size of poly-A tail is usually around 200 A residues, and its role is to protect pre-mRNA from degradation and facilitate the export of mRNA from the nucleus into the cytoplasm. So, what is splicing? If you look at the illustration on the left, you can see that there are three exons, in the molecule on the in the transcript on this illustration, and there are two introns. During splicing, introns are cut and removed, and exons are spliced together. It can be seen on the right panel illustration, where you can see two exons separated by an intron, and the structure called spliceosome essentially cuts that intron out and stitches exons 1 and 2 back together. So let's talk about it a little bit more. The biological importance of the presence of introns, non-coding parts of a gene, is unclear. However, these introns, as we mentioned before, must be removed. Splicing, the removal of introns and stitching back exons, occurs in the nucleus and is extremely sequence specific. The specificity ensures that the introns are properly removed and the exons are joined back together. The 5' end of an intron is marked with the GU sequence, while the 3' end is marked by the AG sequence. The complex of proteins and RNA molecules, a spliceosome, recognizes the ends of an intron, brings them together, cuts the intron out in so-called lariat mechanism, and stitches two exons back together. Now, ribosomal RNA and tRNA, they usually don't undergo splicing because they are not protein encoding. They're synthesized as the long precursor molecules and then undergo cleavage into individual tRNAs or ribosomal RNAs with a subsequent methylation to stabilize the molecule. Now it's time to talk about the process of translation. So proteins are essentially the main workhorses of the cell. They perform almost all cellular functions, and they are synthesized during the process of translation. An mRNA is decoded to form polypeptides. Polypeptides can be of different lengths, from 50 to 1,000 amino acids. And in these polypeptides, amino acids are bonded by a covalent peptide bond between two adjacent amino acid residues. You can see in this illustration that the amino group of one amino acid forms what's called an amide bond with the carboxyl group of another amino acid. The reaction of the peptide bond formation is facilitated by ribosomes, and you can see ribosomal structure on the illustration on the left. You can acknowledge that it consists of a large and small subunits with 3 different sites within the large subunit. And the bottom picture illustrates essentially the process of translation. Transfer RNA bringing in amino acid residues in a specific manner, and the polypeptide that is generated by a ribosome. So ribosomes are a complex of structural and catalytic ribosomal RNAs and multiple polypeptides. Ribosomes are synthesized in the nucleolus, within the nucleus, in eukaryotes, and exist in a cytoplasm and rough endoplasmic reticulum in eukaryotic cells. In prokaryotes, though, ribosomes are located in the cytoplasm because prokaryotes lack membrane-bound organelles. Each ribosome consists of 2 subunits, the large and the small. These subunits are dissociated when translation doesn't happen, but when translation is happening, these subunits associate. In prokaryotes, small subunit has a sedimentation rate of 30 Svedbergs, while large subunit has a sedimentation rate of 50 Svedbergs. You need to remember that Svedberg, the unit that describes the rate of precipitation, is not additive, which means 30s ribosomal unit and 50s ribosomal unit, when they assemble into a prokaryotic ribosome, the sedimentation rate for the ribosome will be 70s. In eukaryotes, the small subunit is larger, 40 Svedbergs. The large subunit is 60 Svedbergs. When they are combined together, the ribosome has the sedimentation rate of 80 Svedbergs. The functions of small and large subunit are quite distinct. Small subunit binds the mRNA, while large subunit binds transfer RNAs and synthesizes a polypeptide. As in eukaryotes and in prokaryotes, mRNA is translated at the same time by many ribosomes. We've already encountered that phenomenon. It's a polysome, and the translation occurs from 5' to 3' end of mRNA. Polypeptide is synthesized from the N to C terminus. You may remember this terminology from our conversation about amino acids. If this is the molecule of polypeptide, on the N terminus, there's going to be an amino group. On the C terminus, there's going to be a carboxyl group. So you can see on the illustration to the left that N terminus comes out of the ribosome first, and C terminus stays attached to the tRNA. What is T or transport or transfer RNA? tRNA molecules are structural RNAs which are synthesized by RNA polymerase III in eukaryotes. Different species have different types, different number of types of tRNAs, and the number of types can reach up to 60. The essential function of tRNA is to carry a specific amino acid and to recognize mRNA codons. In the illustration on the left, you can see the part of tRNA which comprises the attachment site for amino acid phenylalanine. And you also can see the anticodon, the part that recognizes the codon in mRNA. When amino acid is covalently bound to tRNA, this complex is called aminoacyl tRNA. This aminoacyl tRNA binds to the ribosome and allows it to add the amino acids that correspond to the specific mRNA sequence to the polypeptide chain. Essentially, we call this process translation because ribosomes translate the language of nucleotides, the language of RNA, into the language of amino acids, the language of protein. We mentioned before that out of 64 codons, 3 are stop and 61 codons encode amino acids. The codon AUG also can serve as the start codon to designate the place of translation initiation. In each tRNA molecule, there are 3 nucleotides called an anticodon. This anticodon in tRNA pairs with 1 or more codons in mRNA, allowing tRNA and a ribosome to insert an amino acid in the sequence-specific manner. So, for example, the codon for leucine in mRNA is CUA. This codon will bind a tRNA that carries an amino acid leucine, which contains the tRNA which contains a complementary GAU anticodon. You can see for yourself that GAU is complementary to CUA. So at the end of the day, we can say that tRNA has to interact with 3 factors to become functional. First, it must be recognized by the correct enzyme, aminoacyl synthetase, which will place an amino acid onto the tRNA amino acid attachment site. tRNA must be recognized by ribosomes, and the anticodon in the mRNA must recognize a correct codon in mRNA. So again, I want to highlight this. If this cross represents tRNA, it has anticodon. What I cleared out, it was a typo. And anticodon will form complementary pairs with a codon in mRNA. how is amino acid added to the molecule of tRNA? Well, when tRNA is synthesized by RNA polymerase III, only RNA portion of this molecule is produced. Amino acid that corresponds to the anticodon designated amino acid is added later after tRNA is processed and exported from the nucleus. An enzyme called aminoacyl tRNA synthetase, what we call, charges tRNA molecule by attaching an amino acid. Each amino acid is attached by its own unique aminoacyl tRNA synthetase. However, the exact number may vary by species because in some cases there are more than one aminoacyl tRNA synthetase for a single amino acid. Here's what happens. Aminoacyl tRNA synthetase binds ATP and hydrolyzes it to adenosine monophosphate and 2 phosphate residues. Simultaneously, it binds an amino acid and the corresponding tRNA. The energy that aminoacyl tRNA synthetase derives from ATP molecule is invested into formation of high energy bond between an amino acid and its tRNA. This high energy bond, the energy in that bond, will be used later to form a peptide bond during the process of translation. What is happening during the initiation of translation in both pro and eukaryotes? In E. coli, the initiation complex consists of the 30S subunit, mRNA, and initiation factors, as well as initiator tRNA. In order for 30S subunit to bind to mRNA, it recognizes so-called Shine-Dalgarno sequence that is located upstream of the first AUG codon. You may remember that AUG is a start codon and also encodes methionine. 30S subunit gets anchored at the proper location, and this binding of 30S subunit requires initiation factors. Then initiator tRNA binds the start AUG codon. In the illustration on the bottom, you can see the initiation tRNA with a UAC sequence that is complementary to UAG codon, the small ribosomal subunit, and an mRNA molecule. And on the right, you see almost completed initiation complex. tRNA, start codon, mRNA, and small ribosomal subunit. Now, let's go back to prokaryotic translation. Prokaryotic initiation tRNA carries methionine that is modified by formylation, specific type of chemical modification. This is why we call it tRNA-F-met. Formylmethionine, which is not the normal amino acid, is usually removed after translation from the beginning of the polypeptide chain. When the initiation complex is formed, it is joined by the 50S, or the large ribosomal subunit, and that forms competent translation complex. In eukaryotes, the process is similar. Initiation complex is comprised of mRNA, 40S subunit, a variety of eukaryotic initiation factors and molecules of GTP and or ATP. Unlike in prokaryotes, methionine on the initiator tRNA is not formulated. So what is the difference between the initiator tRNA and others methionine tRNAs? Turns out that methionine tRNA that serves as the initiator can bind initiation factors, while other methionine tRNAs cannot. The process of recognition is also different in eukaryotes. Initiation complex, for example, recognizes the 5' cap. For that recognition, so-called cap-binding protein and other initiation factors are required. Just like in prokaryotes, initiation complex in eukaryotes moves along the mRNA from 5' to 3' end towards the start codon AUG. Now, what determines which AUG codon is actually the start codon? It's called Kozak's rules. Certain nucleotides around AUG indicate if it's going to be a start codon. So essentially, the nucleotide environment, the context, determines whether a particular AUG codon is a start codon. Now, this AUG may or may not be the first one. But when ribosome reaches the start codon, the initiation factors and the cap-binding protein dissociate, and 6TS, or large ribosomal subunit, binds to the complex instead. So how is protein synthesized? In other words, what happens during the elongation? The process of protein synthesis is similar in both pro and eukaryotes. Here we're going to illustrate the translation elongation using prokaryotes as an example. So the binding region of tRNA that's located in the large subunit consists of 3 compartments: the A site, the P site, and the E site. A stands for aminoacyl, showing that this is where tRNA binds. P stands for peptidyl, showing that this side binds, that should be corrected actually. It binds charged tRNAs, yes, that is correct, but also this is where polypeptide is synthesized. And finally, the E site is the place where tRNA without an amino acid is released, so that these tRNA molecules can be recharged and come back for the future synthesis. At the beginning of elongation, the initiating formylmethionine tRNA is located at the P site. Ribosome starts moving along the mRNA, and the codons enter the register, enter the A site, and then corresponding tRNA, which has the matching anticodon, binds to the codon in mRNA. So charged tRNAs can enter and exit ribosomes, and this is how new amino acids are added. Energy for each step is provided by elongation factors that break down ATP and release energy. Now, let's take a look at this process a little closer. So in the A site, on the illustration to the right, you can see the charged tRNA. In the P site, you can see the charged tRNA with an entire polypeptide attached to it. During the formation of the peptide bond, the amino group of the amino acid in the A site interacts with carboxyl group of the amino acid in the P site, and this process is facilitated, is catalyzed by the enzyme called peptidyl transferase. What's interesting about that enzyme is that peptidyl transferase is actually a ribosomal RNA, not a protein. So what provides the energy for linking, forming that peptide bond? Energy for peptide bond synthesis, as you may remember, comes from the energy stored in the bond between an amino acid and tRNA itself. When bond is formed, the ribosome shifts, so the tRNA that is now in the A site with polypeptide moves to P site, and the empty tRNA ends up in the E site, where it is expelled from the ribosome for the reusal. Termination of translation occurs when a stop codon enters the A site. You can see here the stop codon enters the A site. Stop codon is recognized by so-called release factors, proteins that are structurally similar to tRNAs. These proteins stimulate the hydrolysis of the bond between a polypeptide and the tRNA within the ribosome, and the release of the polypeptide out of the ribosome. After that, small and large subunits of the ribosome dissociate. They separate from each other, and they separate from mRNA. These subunits usually form another translation initiation complex almost immediately, and at the end of transcription, mRNA is degraded. That allows to regulate the amount of protein in a very precise manner. Nucleotides from that degradation can be further reused in the RNA synthesis in the future. When translation is over, proteins can be further chemically modified. For instance, various signal sequences can be added, and the signal sequences will direct the proteins to specific compartments that will be recognized by so-called signal recognition particles, the proteins that interact with SRP receptors, forming a translocation complex that then directs the protein to wherever it is supposed to be, for example, into the lumen of endoplasmic reticulum. A protein must be folded into a three-dimensional structure. Some proteins can fold by themselves. Some require the helper molecules called chaperones. So for the most part, after the synthesis of protein on the ribosome, the proteins are transported into the lumen of endoplasmic reticulum, where they can be packaged into the vesicles to be secreted from the cell, or they can be further modified, or they can be transported to a different destination inside the cell. Gene expression must be tightly regulated. This regulation is vastly different between pro and eukaryotes. In prokaryotes, there is no membrane-bound nucleus, which means DNA is found in the cytoplasm. It means that transcription and translation can occur almost simultaneously. Another reason why this can happen is that prokaryotic mRNA requires minimal post-transcriptional processing. In eukaryotic organisms, however, there is a nuclear membrane, so the DNA is within the nucleus. The transcription of DNA into RNA molecule occurs before the protein can be formed. Transcription takes place in the nucleus, producing pre-mRNA, which then has to be processed in the process of splicing, capping, and polyadenylation, so the mature functional mRNA can be formed. This mRNA then must be exported from the nucleus into the cytoplasm or endoplasmic reticulum, where it undergoes translation, during which a polypeptide is synthesized. You can see that in eukaryotes, transcription and translation are clearly separated in both time and space. Gene expression in eukaryotes can be regulated at epigenetic, transcriptional, post-transcriptional, translational, post-translational, and nuclear shuttling or export levels. In the future, we're going to talk about the regulation of gene expression in pro and eukaryotes. One of the most famous mechanisms for the regulation of the gene expression in prokaryotes is operons. We're going to talk about tryptophan and lactose operons as perfect examples for the repressible and inducible operons, respectively. Bacteria E. coli can either receive tryptophan from the environment by ingesting it, or it can synthesize it if it's not present in the environment. There are 5 enzymes that are required for that synthesis. 5 genes that encode these 5 enzymes are located next to each other in what's called a tryptophan operon. These 5 genes are transcribed into a single polycistronic mRNA, which is then translated, producing all 5 enzymes. When tryptophan can be found in the environment, the tryptophan operon must be off, so the cell does not waste precious resources. When environmental availability of tryptophan is low, tryptophan operon turns on, allowing cell to supply tryptophan for protein synthesis. Operon can be divided into 3 large regions: the coding region, or structural genes region, which contains the genes for the enzymes; the operator region, which binds tryptophan repressor; and the promoter region that binds RNA polymerase. When tryptophan is present in the cell in the large amounts, the molecules of tryptophan bind to the repressor protein. Repressor, when the tryptophan is bound to it, binds to the operator sequence and physically blocks RNA polymerase from binding to the promoter and transcribing structural genes. However, when tryptophan is not present in the cell, there is no tryptophan bound to repressor, so repressor dissociates from the operator. This leaves no obstacle for RNA polymerase to transcribe the structural genes, and the proteins are synthesized. In this case, repressor acts as a negative regulator, and it keeps the genes off. Since tryptophan operon is negatively regulated, it is also called a repressible operon. Before we start talking about lactose operon, let's talk about so-called catabolite, a little typo here, activator protein. Catabolite activator protein is an example of a positive regulator. The positive regulators bind to DNA and help to turn genes on in so-called inducible operons. So bacteria E. coli can use lactose as the source of energy when its glucose supplies are limited. However, to do this, genes must be transcribed. Now, how does catabolite activator protein assists cells with that? When glucose levels in the environment drop, it results in the accumulation of cyclic AMP within the cell. Cyclic AMP is a special type of a signaling molecule. When cyclic AMP levels go up, cyclic AMP binds to catabolite activator protein, and this complex in turn binds to the cap binding site upstream of the promoter in the lactose operon. Catabolite activator protein bound to cyclic AMP stabilizes RNA polymerase and increases transcriptional efficiency. You can ask yourself, what happens if cyclic AMP levels are low and cap doesn't bind to its binding site? In this case, transcription will still occur, but the rate of it will be quite low. So as we mentioned before, lac operon contains the structural genes that encode enzymes essential for the lactose processing. One of these genes, LAC-Z, encodes the enzyme beta-galactosidase. Beta-galactosidase breaks down lactose, which is a disaccharide, to monosaccharides glucose and galactose. So in order to activate the lac operon, two conditions must be met. First of all, the glucose must be very low or absent. Lactose, on the other hand, must be present. As we mentioned before, when glucose is absent, levels of cyclic AMP go up. Cyclic AMP binds to catabolite activator protein, forming the complex which binds to the promoter of the lac operon and increases transcription. When lactose is present, it is converted into its isomer, so-called allolactose. Allolactose binds to the repressor that sits on the operator sequence in the absence of lactose. When allolactose binds to the repressor, it dissociates from the operator, allowing transcription, allowing RNA polymerase to proceed. So what happens when glucose is present? When glucose is present, the levels of cyclic AMP are low, cap doesn't form a complex with cyclic AMP and doesn't bind to the promoter. When cap doesn't bind to the promoter, transcription efficiency is very low. Now, what happens in the absence of lactose? When lactose is absent, the levels of allolactose go down, which means in the absence of allolactose, the repressor binds to operator and blocks RNA polymerase from proceeding to transcribe structural genes. In other words, lac operon is active only when glucose is low and lactose is high. What if both glucose and lactose are present in the environment? Well, in this case, transcription still occurs, it's just low, since the catabolite activator protein is not bound to the promoter. This table summarizes what happens with the cap and the repressor when glucose and lactose are either present or absent, and how it affects the efficiency of transcription. The regulation of gene expression in eukaryotes is more complicated and occurs in virtually all steps of the protein synthesis. As you just saw, in prokaryotes, the regulation of protein synthesis is for the most part transcriptional. Now, what about eukaryotes? Well, first of all, eukaryotes have the ability of epigenetic regulation. That includes modifications of DNA, acetylation of histones, methylation or demethylation of DNA. Transcriptional regulation involves a variety of transcription factors that determine when and how often RNA is synthesized, in other words, how many RNA molecules will be produced. Post-transcriptional regulation includes RNA processing, RNA export into the cytoplasm. Translational regulation involves the efficiency of RNA synthesis by the ribosomes. And post-translational regulation includes chemical modifications to the proteins and, in some cases, their degradation. So what is epigenetic regulation? Well, first of all, we need to remember that within the nucleus, DNA is wound, folded, and compacted around histone proteins, forming a nucleosome. You can see 8 histone proteins and the DNA molecule wrapped around it. Effectively, nucleosomes control the access of transcriptional machinery to the genes. Whether DNA is bound to histones is regulated by various chemical signals, functional groups, on both histones and the DNA molecule. Chemical modifications to these molecules will determine whether a particular chromosomal region can stay open and available for transcription, or is it going to be closed. These modifications may be added or removed by a set of special enzymes. The most common modifications are phosphorylation, addition of a phosphate group, methylation, addition of the methyl group, or acetylation, addition of the acetyl groups. These groups can be attached to various amino acids in histones. When histone is modified, it alters the charge of the protein. I also want to highlight that DNA sequence itself isn't altered during epigenetic regulation. So the reason why DNA interacts with histones so effectively is that DNA is negatively charged and histones are positively charged. Chemical modifications, such as phosphorylation, can change, or acetylation can change the charge of the histones, which inevitably results in the change to how DNA is packaged around them. For instance, histone acetylation reduces the positive charge, and when positive charge is reduced, the DNA binding to histone also goes down. When DNA isn't tightly bound to histone, that part of the chromatin becomes open to transcription. Another chemical modification, this time modification of the DNA, which also doesn't alter the sequence, is methylation. Methylation mostly occurs at CpG islands, stretches of pairs of cytosine and guanine that surround promoters. When cytosine of CG pair is methylated, the genes are silenced. Methylation can be affected by a variety of environmental conditions and change the gene expression in response to these environmental conditions. If a stretch of DNA is hyper methylated, and the histones are deacetylated. Both of these factors determine that this part of the DNA is tightly wound around histones and therefore transcriptionally inactive. Epigenetic changes, such as methylation or acetylation, are not permanent. However, they can persist through the process of cell division and even can be passed to offspring. If a gene has to be transcribed, the histones and DNA are modified. DNA can be demethylated, while histones will be acetylated. You can see here in this illustration that deacetylated histones are tightly packaged DNA around them, but acetylation allows the DNA to become accessible for transcription. When histones are acetylated and DNA is demethylated, promoter opens so that RNA polymerase and transcription factors can start transcribing the genes. If the gene is to remain silenced, DNA will be methylated, histones deacetylated, and that part of the DNA will wrap itself tightly around histones and stop the transcription. In other words, when chromatin is closed, RNA polymerase and transcription factors cannot bind to the promoter, and transcription cannot occur. As we mentioned before, the core promoter region in eukaryotes is located 25 to 35 bases upstream of the start of transcription. It contains, just like in prokaryotes, Tata box, which binds transcriptional factor to D, Tata binding protein, and then other transcriptional factors are recruited, such as TF2B, TF2E, and so forth. These transcriptional factors facilitate the binding of RNA polymerase to the promoter and activate the initiation of transcription. There are additional elements called promoter proximal elements, which are located within a few hundred base pairs upstream of the start site of transcription. For example, CAAT box or GC box bind specific transcription factors that further increase the efficiency of transcription. Eukaryotic cells have hundreds of different transcription factors, and each of them binds to a particular DNA sequence, which makes the eukaryotic transcription highly regulated. When these transcription factors bind close to the encoded gene upstream, this element in DNA which facilitates the binding of these transcription factors is called a cis-acting element, which means it is located not only close, but also on the same chromosome. Some eukaryotic genes have other regions pretty far upstream from the promoter that increase the efficiency of transcription, called enhancers. Enhancers, while usually are upstream, they don't have to be there. They can be found within the coding region, they can be found downstream the gene, or even far away from the promoter. Specific transcription factors bind to the enhancer region, which results in a change of the shape of the proteins that are bound to the enhancer. Thus, these proteins can interact with a promoter. You can see here that activators that bind to the enhancer can bind to mediator and transcription factor proteins forming a very complex arrangement, highly efficiently regulating eukaryotic transcription. In order for those activators to bind to the transcription factors on the promoter, as you can imagine, DNA must be bent. In order for it to be bent, special DNA bending protein brings the activators closer to the transcription factors bound to the promoter. Proteins that regulate transcriptional efficiency may not only be activators, they may also be repressors. Repressors can also bind to the promoter or to the enhancer and block transcription. Whether or not a certain repressor or activator will be present in the cell often is determined by external stimuli. When transcriptional activators or repressors are produced in response to those external stimuli, that alters the rate of transcription and therefore gene expression. Post-transcriptional modification in eukaryotes involves mostly splicing. Remember, if RNA in eukaryotes isn't processed, protein is not going to be synthesized. So during splicing, as you remember, introns are removed. Well, when exons are stitched back together, there can be some variations in that process. We call it an alternative splicing. In alternative splicing, a single gene can generate different protein products, essentially by generating different combinations of exons. How frequent these splicing alternatives will be produced, or where they're going to be produced, is controlled by the cell. That enables the cell to have different protein products with different functions, albeit relevant to each other, but similar functions, in different cells or at the different stages of organismal development. You can see on the image on the right that there are 4 exons. During alternative splicing, an exon can be skipped, or exons can be mutually exclusive, so either this or this exon will end up in the final product. 5' donor sites during splicing can be different, or 3' acceptor sites can be different. Or an intron can be retained within the sequence. It's important to highlight that the order of exons from 5' to 3' end of mRNA molecule is always conserved. So transcript, which exons numbered from 1 to 7, can only produce alternative splicing variants which are missing 3 or 4 and 5, but they cannot be rearranged in the other direction. Now we mentioned that the start and the end of the intron are highly specifically recognized by the spliceosomes. If these sites contain mutations, that may lead to the skipping of introns. Intron skipping results in various genetic diseases. In many cases, it's an abnormal splicing rather than the variations in the coding sequence. However, alternative splicing provides also a very beneficial trait to the cells, ability to produce new proteins without losing the original version of a protein, and these new proteins may be useful, may facilitate the better adaptation and confer evolutionary advantage to the cell. Another mechanism by which eukaryotes regulate gene expression is the control of stability of mRNA. Enzymes called exonucleases 5' and 3', which shows from which side of the mRNA molecule they will break it down, can degrade RNA by slowly, or not slowly, taking out nucleotide one by one. To protect RNA from exonucleases, it is post-transcriptionally modified by capping and polyadenylation. 5' cap is added to the 5' end of mRNA molecule, while poly A tail is synthesized at the 3' end. 5' cap and poly A allows eukaryotic cells to regulate the lifespan and the decay rate of mRNA, which in turn will affect the amount of protein produced during translation from the specific mRNA template. If RNA has a really high decay rate, its stability will go down and the time available for translation will decrease, resulting in less protein product. If decay rate goes down, mRNA stability increases, which increases the time available for its translation and therefore the amount of protein product. Each mRNA in eukaryotes contains so-called untranslated regions that can be located on 5' and 3' ends. 5' region, as you can see, is located just before the coding region, and 3' region is located downstream of the coding region towards the 3' end of RNA. These untranslated regions can interact with so-called RNA binding proteins, which then can regulate the stability of mRNA, its localization, and even the process of translation. Another mechanism to regulate the stability of mRNA is microRNA. MicroRNAs are short RNA molecules which are produced in the nucleus as longer pre-microRNA. The protein complex called dicer breaks down pre-microRNAs into mature microRNAs, which then will recognize specific sequences in mRNA and bind to it. If the binding of such microRNA is perfect, this microRNA is called SI, small interfering RNA. It will bind the protein complex called RISC, RNA-induced silencing complex, which then lead to mRNA cleavage. However, if microRNA binding to mRNA is imperfect, while RISC will also bind to the target, it will not break down the mRNA molecules, but rather stop the translation from happening because RISC protein is stuck on its target. Now, what about translational and post-translational modifications? Initiation of translation is controlled by the binding of initiation methionine tRNA to the 40S ribosome and initiation factor 2 together with it, or EIF2. As a matter of fact, several initiation factors can bind to 5' cap or poly A tail. That often results in the looping of mRNA molecules within the eukaryotes. Now, as you remember, ribosomes will move along mRNA until they find the start codon, and then when initiator anticodon and the start codon are aligned, initiation factors are going to be released and translation will go into the elongation phase. Now, the binding of this initiation factor 2, EIF2, is controlled by phosphorylation. If initiation factor 2 is phosphorylated, translation is blocked because initiation complex cannot form properly. However, if EIF2 is not phosphorylated, the initiation complex forms normally and translation proceeds. And finally, proteins can also be chemically modified. They can be methylated, phosphorylated, acetylated, or ubiquitinylated. This modification can regulate the activity of the proteins, their half-life, or where they're localized within the cell. Modifications usually occur in response to some kind of an external stimulus, such as stress, starvation, or UV light exposure. The changes, chemical changes in the protein, in turn, can alter epigenetic accessibility of DNA, can alter transcriptional activity, stability of mRNA, or translation if the modified proteins participate in those processes. All of this changes the gene expression. The multitude of regulatory mechanisms allows the cell to rapidly respond to the changes in the environment by quickly altering the levels of specific proteins produced during gene expression. One modification that kind of stands out here is ubiquitinylation. Ubiquitin group is added to the protein and marks it to degradation. Once the protein is ubiquitinylated, it will be bound by a proteasome, which will break down ubiquitinylated proteins into individual amino acids. Ubiquitinylation essentially allows the cell to control the half-life of a protein.